Good afternoon. Welcome, everybody. Thanks for persevering there. I'm delighted to welcome Rick Young as our next speaker in the Broad MIT Seminars in Chemical Biology. Rick is a professor of biology at MIT and a long-standing member of the Whitehead Institute. He has a long list of awards. Just a few to point out. He's been elected to the National Academy of Sciences in 2012 and in 2019 to the National Academy of Medicine. Rick has played an instrumental role in our fundamental understanding of gene expression and how genes are regulated over many years. And in the last few years, he's really uncovered a very pivotal role for phase-separated biomolecular condensates in how genes are expressed. This, of course, has a huge impact on chemical biology if we think about how small molecules are partitioned between these condensates. So I think this is, as we were discussing earlier, a topic that spans biology, chemistry, physics. It's perfect for MIT. So with that, please welcome me and join me in welcoming Rick Young. Thanks.
Thank you so much. It's a real pleasure to talk to folks here at MIT about a subject that I've really become quite excited about. And I've divided this into 4 areas. I want to just briefly introduce how I think about biomolecular condensates, tell you how condensate models have really revolutionized my understanding of gene expression, talk a little bit about condensate-associated diseases and their mechanisms, and then what I hope is especially of interest to this crew, small molecule therapeutics and how we're thinking about the chemical grammar of condensates. So I've been at MIT now for over 30 years, and the way I've taught compartmentalization of cells is largely summarized in this slide. In fact, one of the 2 courses I teach here at MIT is the cell biology of the nucleus. So here you are. Here's the nucleus. Most of what we've taught about compartmentalization involves these membrane-bound organelles. But what's become clear is that the vast majority of compartmentalization that goes on in cells is a transient dynamic phenomenon that likely involves every regulated process that we understand from the perspective of genetics and biochemistry for the last 50 years. Here's an image from John Henninger of the transcription apparatus forming and dissolving at genes as they're activated and then shut down. And as I said, by and large, the literature since about 2017 has come to make us understand that most regulated biological processes in cells are occurring at least during part of their activity in a condensate form. And so these are involved in large-scale biological issues such as cognition and immunity. In fact, the whole reason you can understand anything I'm saying is due to a condensate called the postsynaptic density that behaves in every way that we currently understand as a condensate that's involved in transmitting neuronal impulses. So it's worth thinking just a minute about what it is about proteins initially, one of the 3 major polymers we study as molecular biologists. What features of proteins are contributing to the formation of these dynamic bodies? And I've summarized these on this slide. They come down, they can be classified in 2 ways as low complexity sequences that are forming what we typically call intrinsically disordered regions. These are regions of proteins that tend not to form a stable structure, at least as a monomer, and on occasion will form with a partner a stable structure, but we think within condensates retain a very dynamic kind of structural behavior. There are also these modular interacting domains I've summarized down here. They've long been studied by biochemists and have been known to have the feature of low affinity interaction, but high valency. And that's the characteristic we think about proteins when we think about their ability to interact dynamically with one another. Low affinity, high valency. So the key features I'd like you to think about when you think about condensate formation are summarized in this slide. So condensates are currently thought of as forming through phase separation. It's a property we can examine in vitro with simple systems, but we can only infer in the complex non-equilibrium environment of the cell. Nonetheless, our observations, those of many others, suggest that phase separation is the underlying mechanism. So you get this formation dynamically favored when biomolecules have this interaction with low affinity and high valency, and they reach a saturating concentration where condensate formation is favored. These polymers, DNA, RNA, and protein, are all involved in condensates. Work from Krishna Srivanovas and Arup Chakraborty taught us that DNA molecules that can be modeled as enhancers, where transcription factors bind and regulate genes, that what those enhancer DNA fragments are doing is crowding the transcription factors that are binding them to the point of phase separation. So I now think of these regulatory regions of the genome as simply crowding polymers. RNA is a frequent component of condensates, and the charge balance that occurs when you have these negative strands of polymer together with proteins, portions of which have positive charge, are going to enable formation, stabilization of condensates. And an important point about proteins is that most of the proteins encoded in our genome actually specify what appears to be a structured component, a stable structured component, together with an unstructured component. So about a third of the coding capacity of our genome is devoted to that unstructured piece, so that when a condensate is formed, the structured part is still doing its work. It's still binding to specific substrates, binding to partners, but the disordered part, the way we think about these condensates, is giving us this ability to dynamically form these liquid-like droplets within cells and then dissolve. And the last point I'll make about these condensates that I find fascinating, which is that often they will form in the cell and they'll have liquid-like character. Molecules are moving in and out rapidly. It's very dynamic. The thing will dissolve and reform, that will fuse with other condensates that have like components. But should it encounter anything that would cause, say, cross-linking of the polymers within it, then it will tend to form a gel. And then as it moves toward a solid, a glass. And then as a solid, it's irreversible. And so in neurobiology, there is a subfield interested in neuronal dysregulation that thinks that it's this aging of condensates that may be contributing to the neurological deficit that you get with aging and into diseases like Alzheimer's. The last bit I want to say about condensates is they add regulatory properties beyond those that we typically teach in molecular biology. So they have these features where they will help us concentrate reactants. They'll help us segregate components into the right compartment based on what we believe to be is a kind of chemical difference between different types of condensates. They have what is called metastability. They also have these properties of fusion, fission, and And through a process called Ostwald ripening, components in a small condensate can be stolen by a larger condensate with the same material. So they have these really remarkable features that are not typically studied when we think about biological regulation. So I want to turn for a brief period of time and tell you why I'm so excited about this regulatory space, because it has explained mysteries in my field of gene regulation in a way that no other models have been able to explain. So in my field, gene regulation, the way that regulatory elements function at genes that control cell identity, so each cell, a cardiomyocyte in the heart, a neuron, is going to have a specific set of genes that's on that define that cell's identity. And that regulation occurs through cell type specific elements. Of course, the DNA is the same in all our cells, but these elements are on only in the appropriate cell. And it's that DNA is the platform for binding of cell type specific transcription factors. So the transcription factors are in fact defining what DNA is going to be utilized for that cell. And they bind there, they recruit coactivators and the transcription apparatus, and they become a target for signals outside the cell. So the way most signals outside the cell are recognized to cause the cell to change the gene expression program is they are brought in first through a receptor in the plasma membrane, they transit through the cytoplasm into the nucleus, and they find their way exactly to these enhancers that are controlling cell identity. How that happens, I think, is all wrapped up in condensate biology. So let me tell you about 3 mysteries that have been mysteries for really nearly half a century that I think are nicely addressed by condensate models. So the first is your transcription factors. You have about 1,600 encoded in your genome. 300 to 400 of these are expressed, they're transcribed in each cell. And an important subset of these are these enhancer binding transcription factors. All the transcription factors have a DNA binding domain that is structured. It's stable. You can make the protein as a recombinant and assemble it with a partner in vitro with nothing other than a segment of DNA, and it will retain a stable structure amenable to crystallography. And so all of the structures we know of dozens and dozens of transcription factors, involve these DNA binding domains and the specific kinds of sequences they bind. But they all have an effector domain, often called an activation domain, and that's completely disordered. That has never formed a structure stable enough to capture in crystallography. Not one. Not one activation domain of any transcription factor in any animal has ever been crystallized and had its structure solved. But this is the business end. This is what does the work of activating the gene. So how could it be that we've known about these things for half a century, but we don't understand how they work? Another mystery, about a decade ago, my lab, collaborating with Phil Sharp's lab, and a lab over at Dana-Farber run by Jay Bradner, who became the president of Novartis Institutes, we discovered that genes that are playing the most prominent roles in cell identity, genes, for example, that encode the transcription factors that can reprogram cells from one identity to another, those genes almost always have clusters of enhancers. And we didn't understand why, but we called them super enhancers. And we knew they had a number of properties. We knew that they're occupied by a very high density of transcription factors. They have a very high density of what are called enhancer RNAs. RNAs are transcribed from enhancers. We knew from DNA interaction data that each of the enhancers that are clustered tends to interact with one another. They tend to come together into foci. And bizarrely, although the conventional model for how gene regulation occurs is that the enhancer-bound factors loop to the promoter of the target gene, in many cases, these super enhancers only needed to come within about 150 nanometers of the target gene to activate it. They didn't need to come touch it. So that was a mystery. And the third mystery I want to point you to are roles for RNA and transcriptional control. I mentioned enhancers are transcribed, but we haven't known what those RNAs do. And moreover, we know that when transcription occurs, it's not continuous. It's discontinuous. We call it bursty behavior. So rather than getting one transcript per hour for 24 hours, if we needed 24 transcripts, we just make 24 transcripts right now and then silence the gene for the rest of the day. How that happens has not been understood. And it was in about 2009, it was exactly in 2009, that Tony Hyman, a cell biologist, with Frank Ulicker, a theoretical physicist, observed with their graduate student, Cliff Brangwine, that a body called P granules in germ cells had liquid-like properties. And this is the first time that such a thing had been described in cells, and it inspired others to begin looking at whether or not there were non-membrane encapsulated bodies that look like liquid droplets elsewhere. And one of the groups that was inspired was my own, together with Arup Chakraborty here and Phil Sharp. And we thought maybe some of these problems I've just described to you would be addressed if, in fact, condensates were formed, if instead of having one molecule of RNA polymerase recruited to genes with one molecule of a co-activator, we had dozens of molecules forming an assembly that wasn't stoichiometric, that was governed by other properties. And Arup's group, Krishna Srinivas, developed a math to describe this, and we thought we should set about doing some experimental tests. Sure enough, using microscopy, we were able to go look in embryonic stem cells. These are cells that are essentially identical to the cells in the blastocyst that's being implanted in very early embryogenesis, and that are what we call pluripotent. They're capable of forming any cell in the adult. And they have a key transcription factor called OCT4, which we could monitor by immunofluorescence. And this is the large subunit of the coactivator that operates at all these enhancers. So coactivators, by definition, are factors that don't bind DNA, but are essential for the transcriptional process. And this mediator protein does exactly that. It mediates the interaction between the transcription factor and the RNA polymerase apparatus. And here we're using FISH, so we're monitoring where the DNA is. And this is the NANOG gene. NANOG encodes one of the pluripotency transcription factors, and it has a super enhancer. And so what we're seeing here is that OCT4 and MED1 were co-occupying the super enhancer. And this was just static data to show you that what we're looking at a particular super enhancer is actually a pretty large cluster of molecules. We estimated using other techniques that the amount of the mediator coactivator there involved not 1 or 2 or 10 molecules, but perhaps over 100. And similarly, that there are about 100 molecules of RNA polymerase at this site. And importantly, the model predicts that we ought to be able to take the key components, mediator and the transcription factors, and see them interact in a condensate in vitro. So we could purify these components and ask for all kinds of famous transcription factors, whether or not these factors fused to monomeric EGFP would form a condensate with this MED1 coactivator protein. And they did, but more importantly, the amino acids that we know in the transcription factors that are critical for gene activation, when mutated, you get no more gene activation. In all the cases that we examined, those also prevented phase separation. And moreover, in our cells, we have transcription factors called nuclear hormone receptors. So these are the receptors for estrogen, for androgens. These ligands were necessary for the estrogen receptor, a transcription factor, to fuse with and to become part of this mediator condensate. So this was just some of the data that led us to believe that phase separation by these components was involved in the formation of a condensate. Here's a model for that. So here, the red sites are sites of transcription factor binding at enhancers. So you have here 1, 2, 3, 4, 5, 6 enhancer DNA segments bound by red transcription factors. And the mediator components are in blue and polymerase is in green. And these little squiggly lines are representing the dynamics of the disordered regions of those proteins. So again, what you have are proteins that have structured domains. Those structured domains, by conformational change, do their work. But you have these disordered segments that interact with one another in this dynamic fashion. High valency, low affinity interactions to produce, at least for a period of time, this condensate. And in doing work with Ibrahim Sisi here, we found that these are typically about 200 nanometers in diameter. They have a size range, but this would be average. They have about 100 molecules of each of mediator and Pol2. That's very different than any of your textbook models will show. They're dynamic. They exhibit this liquid-like behavior. If we use a laser to photobleach these molecules, we can see how rapidly they're diffusing inside the condensate and with material outside. And it was interesting to see that their size, lifetime, and transcriptional output are correlated. That is to say, the larger these condensates, the more transcriptional output, and the longer we could see them in that movie, which all made sense for a condensate. And the model helped us understand then how these activation domains function, how the enhancers are functioning. They're crowding the transcription factors to the point of phase separation. Now we understand why there's such a dense assembly of transcriptional components that we saw in those gene tracks that we determined by a technique called ChIP-seq. And now we could see why some enhancers, like this one up here, can participate in activating a gene, but not actually get any closer than about 100 nanometers. It was an interesting field in condensate biology that's been around since the 1950s, where chemists would study what they called complex coacervates. Complex because they had two molecules, one negatively charged, one positively charged. And what they found in this field is that through charge balance, you could get an optimal size condensate with optimal stability. So if you take, for example, a protein that has positively charged residues and an RNA with negatively charged residues, and you hold one constant, then as you added more and more, let's say, RNA here, you would eventually balance the charge and form this condensate. But as you increase the amount of RNA, those excess RNA molecules would steal proteins, and you would dissolve the condensate. So this was called reentrant phase separation. And that model inspired John Henniger and Oscar Oxus to get together with students in Arup Chakraborty's lab and test this model whereby the RNA molecules that are being produced at low levels at enhancers might actually stimulate the formation of these transcriptional condensates. But that once you began transcribing, the additional RNA molecules would then contribute to dissolving it. It's a very elegant model because the product of the process is negatively regulating the process. And in fact, we think this is why these condensates form and dissolve, at least one of the features, and also why you get bursty behavior. If you're going to recruit a dozen RNA polymerase molecules or more and have them act back to back like a little choo-choo train, creating lots of RNA density, then that RNA density is going to dissolve the condensate, and you'll get a burst, and it will stop. So this conveniently explains a number of observations in my field. And to finish this part, I'm going to tell you about a number of observations that we've made over the last decade in cancer cells that we finally understand using this model. So one observation is that in metastatic tumor cells, the type that kill you, the driver oncogenes build superenhancers. They evolve superenhancers. But they're not like the ones in our normal cells. In our normal cells, they might occupy 10 to 20 kb of DNA. In a metastatic tumor cell, they can occupy 500 kb of DNA. I've never understood why you need to occupy DNA with so much transcription apparatus, but now I realize that helps create a very large condensate, and they are very long-lived, and now you're delimited for the transcription apparatus. You can just transcribe that oncogene all you want. Another set of observations that baffled me was there are tumor cells where a single point mutation can make a binding site for a single transcription factor molecule, and that will form a superenhancer with hundreds or thousands of transcription factors bound surrounding that. There's some kind of extraordinary cooperativity I've not seen anywhere in molecular biology that must account for this, and when I understood that phase separation might be a model to explain it, then I had a way of thinking about how you get almost a digital response to such a small perturbation. All the developmental signaling pathways that are critical for us to develop from a single cell into the 30 trillion cells, there are three major ones, the Wnt, beta-catenin, and other one, Wnt, beta-catenin, and TGF-beta. And all of those effectors are highly disordered. And what we realize is the terminal components of signaling pathways find their way to superenhancers and other sites where there are large condensates by simply fusing with them. They favor interacting with that chemical environment. So that helped us understand why there was such favored even though these components are expressed in every cell type, they only go to the genes that are specific to that cell identity. So this explained how that could happen in every cell. And then, to a point I'm going to make later on, there was a mystery that emerged about 10 years ago when Jay Bradner and Nathaniel Gray and I were studying transcriptional inhibitors. And new ones, epigenetic inhibitors like JQ1. So the inhibitors that we studied directed to the CDK7 cyclin-dependent kinases critical for one of the steps in transcription initiation, and BRD4 is a chromatin regulator that binds acetylated histones. These are factors that occur at every active gene. But what we saw in one cancer after another was that the genes that were primarily affected by the drug were the driver oncogenes, which is wonderfully fortuitous, but a mystery up until we realized more recently that this is a consequence of these drugs concentrating into condensates. So I want to turn and just briefly talk to you about what we've come to understand about condensate-associated diseases and some of the underlying mechanisms. And as I talked to you, as I mentioned earlier, we think that there's some neurodegenerative diseases that involve condensate dysregulation. And here are three where we think condensate behaviors are contributing. So one is called Rett syndrome. That's where we believe that condensates that are forming in heterochromatin are lost. In a Wilms tumor, we think this is a case where a transcription factor is inappropriately forming condensates. And in Ewing sarcoma, where we think a transcription factor fusion is getting into the wrong compartment. It's supposed to be in the nucleus, but it's out in the cytoplasm. And moreover, to these material properties I mentioned to you early on, in ALS, we suspect there's a liquid to solid transition. And in a developmental phenotype called synpolydactyly, where you change the number of digits, we think there's altered composition to these condensates. And here's some of the references that refer to that. But no one had examined all the pathogenic mutations that affect these condensate-promoting proteins. And so Salman Banani and Lena Fayan and Susanna Wilson-Harkin decided to do this, and they were interested both in pathological human genetic variation and how it might create those effects, but we've also been interested in environmental factors, and I'll mention a new discovery about insulin resistance and type 2 diabetes in this context. So what they did, and this was published recently in Developmental Cell, is they defined a set of condensate-forming proteins. So they basically collected all the proteins that they think are engaged in condensate formation. They defined where within those proteins these modular interacting domains and low complexity sequences occur. And then they annotated these with likely pathological mutations from clinical databases. And then they took these 300,000 pathological mutations mapped to about 30,000 instances of a variation that they thought might affect condensates. And they tested in this murine embryonic stem cell system where they could transfect with a wild-type protein versus a mutant protein and see if they could find some evidence consistent with the idea that the pathogenic mutations were altering condensate behaviors. And nearly 90% of those tested candidates, in fact, displayed dysregulation in this model system. So now they have a catalog they've published. You can find this catalog online. They think there are pathogenic mutations that contribute to at least 1,700 diseases. I think it's actually far more, just because of the method we're using here. We're only focused on these disordered sequences and not really focused on the extent to which mutations in the structured portions of proteins could also influence condensate behaviors. So we think this catalog provides a resource for much further work in the area of disease hypothesis. And even more recently, Alessandro Delagnesi and Jesse Platt, together with a huge collaboration with Rudolf Janish's lab and Linda Griffith's lab, with Phil Sharp, have come to a model in which type 2 diabetes, insulin resistance, is influenced by the behavior of the insulin receptor, which is a receptor tyrosine kinase, and which, with insulin signaling, will integrate into a condensate with its substrates. It can be found in the cytoplasm associated with vesicles because there's recycling of the insulin receptor. It can migrate into the nucleus where it becomes associated with genes that are actually insulin responsive. And what's remarkable about what they see in a dysregulated environment in the context of insulin resistance and type 2 diabetes is that in those insulin resistant cells, the same things happen, but the condensates are less dynamic and they're longer lived. They are basically what they have termed lethargic. And that's fascinating to us. The first-line drug that one takes for type 2 diabetes, for insulin resistance, is metformin. Metformin partially reverses this phenotype. So that's how we're thinking that metformin may work in addition to its influence on the respiratory chain in the mitochondria, which influences the level of reactive oxygen species in the cell. And indeed, we suspect that it's high ROS in these insulin-resistant cells that's contributing in some fashion to this condensate dysregulation. So there's no change in the number of insulin receptor molecules. There's a change in their behavior within the context of these condensates. And the most widely used therapeutic is affecting that behavior. Well, I want to finish then by talking to you about the newest area that we're focused on and try and get you to think a little bit about this area and maybe collaborate with us to understand some of the chemistry behind condensates. And this is, the team has been working on this, and at the moment it's really led by Henry Kilgore. So I'll remind you that when we think about the pharmacology of a drug, we think about pharmacodynamics, and we think about pharmacokinetics. And pharmacodynamics is mostly about how the drug acts on us, and pharmacokinetics is mostly how we treat the drug. And absorption, distribution, metabolism, excretion, or the ADME is the acronym for the way we think about pharmacokinetics. But a question for us is, when we think about distribution, we're talking about tissue distribution. We're not really talking about cellular distribution, but in fact, drugs, as far as we can see, do not typically distribute themselves evenly throughout the cell. So Henry and Kellen Overholt have done experiments with a whole range of therapeutic drugs where they've asked for the subset of drugs that are naturally fluorescent. What does it look like when you image them in any one cell under the same conditions? And what you get are images like this. And by and large, the drugs that act on a particular target are going to the right place. So for example, camptothecin, that's a topoisomerase inhibitor, and topoisomerase is important in the transcriptional process at active genes, and so it makes sense that that drug would go into the nucleus. But there's some cases, let's see on here, sunitinib, where this drug is an RTK inhibitor. These RTKs are typically out on the plasma membrane, but that is not where this drug is concentrating. So we wonder to what extent the concentration of drugs in various compartments is influencing their therapeutic activity. And one way to look at this is to create a model system. So we've taken this model system where we're looking at what are called scaffold proteins for the condensates. So biochemists have decided that if there is in a condensate a particularly abundant protein, a protein that interacts with many other proteins, they'll define that as a scaffold, much using the classical definition of the word, it's interacting with many proteins. You take these scaffold proteins and we've taken them from the transcriptional condensates, splicing condensates, heterochromatin condensates, nuclear condensates. And those recombinant proteins will phase separate and produce droplets in vitro. And often you can get this to happen under relatively physiological concentrations of protein and components. What you can do with these droplets then is you can visualize them in an assay where you've added a molecule that is fluorescent. So here we have fluorescein, and what you're seeing here are images with, these proteins are tagged with monomeric EGFP, so you're seeing this green color here, and the small molecule is diffusing through that solution, and it doesn't seem to be concentrating or excluded from any of these condensates, and here's the quantification over here. But by contrast, a drug like cisplatin, normally interacting with double-stranded DNA and to some extent RNA, there's no nucleic acid in these assays as far as we know, but what it's doing is it's concentrating selectively in this MED1 condensate. And here's the quantification. We get some fluorescence quenching in these assays, so in other assays, we've been able to do better quantification, and in the better quantification, we think that cisplatin's being concentrated hundreds of fold inside the condensate relative to the surrounding solution. Well, does that matter in vivo? And if you take a tumor cell and you add cisplatin, and you wait about 6 hours, and what we can do is prepare the DNA, chop it up, and use an antibody that recognizes only platinated DNA, so that we can ask across the genome, where did the cisplatin end up platinating DNA? And it ends up platinating enhancers, but in particular, the super enhancers where we know these MED1 containing condensates occur. So this and other data led us to the following model. In normal cells, which are exposed to cisplatin when you're getting this type of therapy for cancer, the transient transcriptional condensates that are forming may well concentrate cisplatin, but it's going to be a transient effect. By contrast, in the tumor cell, these very large condensates that are forming on the driver oncogene will have the time to concentrate very high levels of cisplatin, which will act on the associated DNA and ultimately destroy that gene. So it's interesting, we've moved from a model where cisplatin just generally damages DNA, and because a tumor cell has to proliferate, the idea is that the damaged DNA takes a little time to get repaired, so you're selectively damaging the tumor cells. I'm sure that's at least partly true. But in this model, what the cisplatin is doing is it is selectively destroying the oncogene itself, the oncogene driver in that cell. I find that very interesting. This is the most widely used, well, the platin family is the most widely used anti-cancer agent in the world. So it may work by a mechanism that's not a textbook mechanism. And if you ask an oncologist what might be the most persuasive evidence for mechanism, they'll tell you, well, it tell me something about drug resistance. And so I find it interesting that tamoxifen, the anti-estrogen that is the primary line of defense in breast cancer, it competes with estrogen's binding site on the estrogen receptor. If you get resistance to tamoxifen, that can occur by two mechanisms. One is classic, amino acid change in the tamoxifen binding site. But the other is not so classic. Those are alterations that simply overexpress the normal amino acids in MED1. And only in a condensate model can I come up with a mechanism whereby overexpression of the condensate-forming protein into which tamoxifen is going to concentrate. Only in that context do I understand how that might work. And patients with this type of resistance have the worst possible prognosis. So I think we're coming to understand, at least in this case, a mechanism of anti-cancer drug resistance that we had not anticipated. Okay, so Henry and Callan have been very interested in figuring out whether or not different condensates have different chemical environments, and whether they can use small molecules to probe that, and whether they can use machine learning with Regina Barzolet's lab to predict small molecules that selectively partition into those chemical environments. And finally, whether such an algorithm could work at all in the context of a living cell. So what they've done is akin to what I showed you earlier. They have taken the proteins that are scaffolding proteins for these three different condensates. They've reconstituted them in these in vitro droplets, so now they can use them as a screening platform. And in order to screen, they have used a library of several thousand BODIPI-containing cores that allow them to do confocal microscopy and determine for each compound its partitioning behavior. And this is just some evidence that the subset of compounds that exhibit the best partitioning in the MED1 droplet are not behaving the same in these other droplets. So the chemical environment of the MED1 droplet appears to be substantially different than that for HP1 alpha and for NPM1. And the reverse is true, the converse is true as well. So these are selective environments. So Peter Michel in Regina's lab has developed a way of examining these structures and putting them through a deep learning model where the model can identify what they call rationales. And that model, we can ask for a prediction about whether or not an untested molecule is likely to partition selectively into that condensate. And that model is remarkably accurate in predicting the next chemical compound's behavior with respect to these condensates. But what we found utterly shocking is that that model is somewhat predictive of what an untested small molecule will do in a living cell. And I say that because I was quite surprised because the condensate in the living cell has hundreds, if not thousands, of different kinds of molecules, protein and RNA, sometimes DNA, and it's in a non-equilibrium environment. We did our initial chemical probing of a phase-separated condensate with a single protein molecule in an equilibrium environment. So how could this possibly be predictive at all? And it is. So we're guessing that what's happening is that the scaffold proteins form little microenvironments that are chemical environments that ideally solvate certain of these molecules, and that those little chemical environments are retained in the more complex, dynamic, and non-equilibrium environment of the living cell. That's one of our hypotheses. So we think there are different chemical environments in different biomolecular condensates. We think the environment of transcriptional condensate and a splicing condensate and a translational condensate are different. We think that we're getting clues to those different environments using these small molecule probes and machine learning, and it is giving us ideas that we hope might facilitate future small molecule therapeutics development. So what have I told you? I've told you that biomolecular condensates are helping us distribute our 10 billion protein molecules in the cell such that they can find their partners in various functions and function in a dynamic and efficient way. That pathogenic mutations, we think, can cause condensate dysregulation in really a broad spectrum of diseases. And that we think that small molecules selectively partition into chemical environments that we can now probe, and that with machine learning can learn what are the various features that make up a machine learning vector that describes the behavior of that chemistry. So I just want to leave you with these three questions. We need to understand in the future, what is this chemical grammar? And you can look at it from both sides of the equation. What are the chemical rules governing the condensate behavior, and what are the chemical rules governing how small molecules and biomolecules interact with that environment? How do non-equilibrium processes influence condensate behaviors? We suspect that equilibrium processes may facilitate their formation, but non-equilibrium processes are going to prevent them from overtaking the cell, are going to regulate, in fact, their dissolution. And we wonder, of course, whether novel therapies can be developed based on these hypotheses. Thank you very much.
Thank you. That was wonderful. I have tens of questions myself, but we're going to open it up. There are some microphones in the back, and anyone online may also type into the Q&A. So give some thought to questions you might want to ask. We'll start with, there is one online already. Does the very first event in the initiation of the condensate formation require any high affinity plus multivalent interaction of the first two partners? Of course, this may be context or partner dependent.
That's an interesting question, and I don't think we know. I would assert, though, that because you can either create assemblies with high affinity interactions or with these low affinity, high valent interactions, that you could initiate in either way. What you're trying to accomplish is a multi-molecular assembly that is not governed by stoichiometry. So initiating it with a high affinity interaction would be fine with me.
Please.
Thank you very much. So I'm really curious about the small molecule screening. Like when you're doing screening, is it like the result, a binary result? It's like it's on a droplet or not droplet. Is there something between? Like if for example, it's still a droplet, maybe the property is like a little bit different.
Okay, so we can create environments in which the formation of the condensate can be digital in the sense that we can reach a saturating concentration of the protein molecule where we initially see the formation of those droplets. But when we are assaying the small molecules, we're looking at the ratio of small molecule in the condensate versus outside.
That was terrific, Rick. To what extent does post-translational modification play a role here? Phosphorylation and methylation of the proteins that are the scaffolds or other proteins that are in condensate, that are in the condensates, can that be used Can controlling post-translational modification help with the control of condensate formation or dissolution?
Yeah, I think actually that's likely to be the major way that cells have evolved to control condensates. And so, for example, we've long had a situation where we knew that RNA polymerase II, when it transitions from initiation to elongation, starts from a state where it has an IDR that's 20 kD of protein and not phosphorylated, to where post-initiation, it's hyperphosphorylated. And what we found was that the unphosphorylated form of polymerase really likes to be with all of its proteinaceous friends in the transcriptional condensate. But when phosphorylated, it's driven out of that condensate and it helps form a splicing condensate right beside that. So we actually think that I study gene expression, so everything from me is about gene expression. But here's the case in our field where phosphorylation plays this dramatic role in initiation to elongation, and it's taking you from that transcriptional condensate immediately into a splicing condensate. And one of the things we've learned about splicing in the last few years is it's not just co-transcriptional, it's happening within 100 to 200 nucleotides of synthesis. So it's happening immediately behind the polymerase. So this is just one line of evidence that makes me think that post-translational modifications, and in particular phosphorylation, is a switch for many reactions.
Great. We have one more online. Given the number of BRD4 degraders that are out there now, what are your predictions on the effect of them on condensate formation?
Yeah, so it's very interesting that there's such a profound difference between small molecules that are acting as competitive inhibitors of BRD4, which is the way Jay Bradner and others initially got into that. This is an anti-cancer therapeutic concept. And then degraders came along with the idea that simply eliminating BRD4 in cancer cells that were highly dependent on it would be a good therapeutic approach. But there's really a striking difference in the behavior of the cell if it has a competitive inhibitor of that acetyllysine binding. And it chases that molecule off, and now you have a change in condensate behavior, transcriptional condensate behavior. If you now just degrade the protein, you might expect a very different effect. So I can't predict whether it's a good thing or a bad thing, but it should be different.
Right, but it's an interesting point to how many, for example, even in the kinase field, kinase inhibitors generally don't have the same effect. An inhibitor might not have the same effect as a knockout, right?
Yep.
For maybe the same reason.
Exactly.
Laura, please.
Hey, thanks for that great talk. I'm curious about the molecule partitioning. And one of the things I noticed with the molecules you showed is they have aromatic groups. And that's actually true of a lot of the kinds of anti-cancer drugs we use. So I wondered if you'd looked at non-aromatic drugs and whether they similarly partition or not, because I think like that aromatic ring might be really relevant for the partitioning.
Yeah, let's see. If Henry's in the audience, there he is. He knows the answer to that question.
Do you?
I do. So most of the compounds we look at least have some content of aromatic ring systems to them, but they're also highly diversified. So there'll also be long alkyl chains, things of that nature, cyclohexyl rings, a wide array of your hydrocarbons and saturated nitrogen heterocycles. And what we found is that those components can modulate the activity quite a bit as well. So I don't believe it is just the case that aromatic rings are the sole thing that's happening here. I think the chemical environments that are being created inside of these bodies can interact with a wide array of different
That wasn't really my question, though. My question was whether or not things that are non-aromatic go into condensates. Because I give you that the environment is going to matter, I'm just wondering if other kinds of molecules go in.
For example, we've done experiments where we've looked at using mass spectrometry-based assays, the partitioning of other small molecules. And in those cases, you do see partition ratios of pretty significant amounts as well.
Of non-aromatics.
Of non-aromatic.
Yeah, not positively charged either, because you know what I mean. Like, yeah. I think that would be interesting. The best.
Of course, in the front.
Phenomena of regulating the composition of condensates to create different behaviors, like more of one scaffolding protein to make it more responsive to RNA feedback or something?
I'm trying to think of a really cute example I can give you of that. But what I'm reminded of is anytime we overexpress a protein, we have this liability, and that is that if it's involved in condensate formation, it can alter the behavior of its normal home. And if for some reason, let's say it's a fusion protein that's abnormal and that fusion stays in the cytoplasm rather than the nucleus, then it will, for example, there are cases where there are factors that are involved in spliceosome behavior that have undergone a mutation, so they've been fused to something that causes them to lose their nuclear localization signal. They will then form a condensate in the cytoplasm using some of the splice apparatus that should be in the nucleus. So I'm not sure that answers your question, but anytime you place a protein in excess or you place it in the wrong place, it does seem to change either the behavior of its normal condensate or it takes that condensate into a new compartment.
In normal functional condensates, the composition is more or less like well regulated and well preserved?
Well, in the normal function of a condensate, we're not yet at the point where we understand all the non-equilibrium features that are in that balance of power, if you will, between the forces that are causing the assembly and the forces that are causing its dissolution. So I'm only answering your question with very high-level words, not with specifics, because I don't think we have the experimental data to address it.
Wonderful. One more in the back. That's you raising your hand. Go ahead with that.
Interactions probably at some degree like non-stoichiometry. So have to think about maybe putting two small molecular into the system to disrupt it and to see if there's any, like combination effect, whether it's like 1 + 1 bigger than 2 or it's like 1 + 1 smaller than 2.
So say again how you want to disrupt it?
Well, like putting two small molecules into the system at the same time, and would this be like a way to solve some like combination effects in therapeutics?
Yeah, I love that idea, and Dr. Kilgore has some evidence that in fact some small molecules will enter a condensate, like one of these mediator condensates, and now change its chemical properties so that it will alter the behavior of another small molecule with respect to its partitioning into that condensate. So you can have these effects where the entry of one small molecule is going to change the behavior of a second one in a quite dramatic way.
Very interesting. Please.
On your slide there about the chemical grammar. So is there selectivity with the RNA sequences in condensates, or is it highly heterogeneous?
It's a really good question. And, in many condensates, the RNA molecules that are there are selective for that condensate. And so you have to wonder how that occurs, whether that's through some high affinity interactions with specific sequences or secondary structures or tertiary structures. You know, the best studied condensates with respect to RNA are these stress granules that just accumulate every kind of RNA. So they're not a good platform to study that question.
But when you make your reconstituted ones,
Yes.
specific sequences, or?
When we reconstitute and we're using RNA, we'll try and use different sequences that would give us some suggestion of whether or not they're sequence-specific behaviors. But you know, the one caution here is that unless it's poly-U, almost any sequence will form secondary structures. And without knowledge of what those secondary structures are, we're a little uncertain about this issue of specificity. There are a lot of secondary structures that are normally recognized by RNA binding proteins, and the specific primary sequence in some of those cases matters less than the secondary structure.
And then with your non-equilibrium process, where you go from gel to solid, you mentioned that there's crosslinks between the proteins. Do you know what the nature of the crosslinks are? Is it just disulfides or something else?
We don't know. In fact, one of the things that we're trying to determine right now is, given that there's high ROS in insulin-resistant cells, if the consequences of that oxidative stress are contributing to some of that immobility, and that's my favorite hypothesis at the moment. I don't know whether that's mediated by the protein or mediated by RNA or other components, but the suspicion is it's protein.
Wonderful. Well, thank you again, Rick. You've given us so much food for thought. And let's all thank Rick for a great talk.